Good morning, everyone. Thank you very much, Giovanni, for your kind introduction. Thank you for the organizing committee to giving me here today the opportunity to present our work. First of all, I have another disclaimer. As Tracy mentioned before, I am a synthetic organic chemist with little experience on Allostery. So we are two outsiders in this meeting, but I hope I will give you some good inspirations for the work we have been done over the last, or chemical biology actually, project we have been working on in the last 3 years, which is part of the broader drug discovery project that we've been working on over the past 10 years, trying to develop or identify small molecule modulators to treat cystic fibrosis. Here we are. So, a brief introduction about the disease. Cystic fibrosis is an autosomal recessive genetic disease that affects primarily the respiratory tract, particularly the lungs, with constant inflammation and infection states due to a thick mucus that, due to the mutations of protein that we show in a while, gets a really thick mucus that brings to these problems. Cystic fibrosis is actually the more common of rare genetic diseases, affecting roughly 1,000 people worldwide, mainly of Caucasian origin. We believe that in Italy, 1 out of 30 person is healthy carrier of CF mutation. So indeed, is not so rare at all as a disease. Despite the great improvements in the treatment of CF patients, cystic fibrosis remains a treatment burden where still unmet medical needs. So getting into the project, so the disease is caused by mutations in this cystic fibrosis transmembrane conductor regulator gene that lead to loss of function and loss of expression of a CFTR protein. CFTR is an epithelial ion channel involved mainly in the transport of anions, primarily chloride and bicarbonate. It's present in multiple organs, so that's why generally cystic fibrosis is defined as multi-organ disease. But as I mentioned before, the major cause of morbidity and mortality of the patients is due to the lung problems. So CFTR is a protein that belongs to the ATP binding cassette transporter family of proteins. Is a glycoprotein made of roughly 1,500 amino acids and consists of different portions, different domains. 2 transmembrane domains, TMD1 and TMD2, and TMD2 here depicted. 2 cytosolic nucleotide binding domains that bind the ATP, which are depicted here, and a unique, with respect to the ATP binding cassette superfamily, a unique cytosolic regulatory domain. This regulatory domain, in its dephosphorylated form, doesn't allow the 2 nucleotide binding domains to get closer, and therefore the PO is in its close conformation. Upon phosphorylation, and the R domain is more structurally organized, and which then leads to the 2 nucleotide binding domains to get closer, to dimerize and open the pore and get the ions passing through. So having said that, there are more than 2,000 known CFTR mutations. Among them, around 400 are CF causing. The mutations have been classified in 6-7 different classes based on their phenotypic consequences, affecting then the synthesis of the protein, the processing, the trafficking, the function, and the turnover. The most frequent mutation is the one which brings to the deletion of the phenylalanine position 508 from now on F508del mutation, which is present at the level of the nucleotide binding domain 1. This is, as I said, the most common mutation present in almost, I would say, around 80% of the CF patients. So, as I mentioned in the first slide, over the past 10, 15 years, a major breakthrough has happened in the life of CF patients due to the start in research and discovery on novel CFTR modulators. CFTR modulators are basically small molecule compounds that primarily address the basic defects of the mutant protein. There are different classes of CFTR modulators. The most studied ones are the potentiators, which mainly improve the opening or the gating of this mutant protein, and most of all, the correctors. Basically, correctors are small molecule compounds that try to improve the processing and trafficking of the mutant CFTR up to the apical membrane. This is an important slide because I will mention this quite often during the presentation. Correctors have been classified according to their postulated mechanism of action in 3 different types: corrector step 1, 2, and 3. Corrector step 1 have been, we believe they interact with the interface of nucleotide binding domain and transmembrane domain 1, so basically this side of the CFTR protein. Type 2 correctors target directly NMB2 here, basically at this side, while type 3 correctors stabilize the portion of the protein where the delta F mutation has been reported. So type 1, 2, and 3. Remember that. Actually, cystic fibrosis is one or probably the first example of personalized medicine, because CF patients have been treated so far depending on their specific mutations with the available drugs at the moment. So patients having type 3 mutations, basically gating mutations, have been treated with the correctors, improving as I mentioned before, the gating, the opening of the mutant protein. Instead, for patients carrying mutations of type 2, so basically affecting the trafficking and the folding on the protein, have been mainly treated with a combination of correctors of type 1, sorry, lumacaftor VX801 and tezacaftor VX601, or as I mentioned, the combination with the type 3 of correctors. So these are so far the drugs available for the treatment of CF patients. So now we go back to 2014 when the Fondazione per la Ricerca sulla Fibrocistica in Italy decided to finance a drug discovery project aimed at the identification of novel drugs. So you can say, hey, why do you need novel drugs? Because taking a step back, all these available drugs have been developed and produced and commercialized by one single company. So in order to somehow differentiate this and improve the quality of the drugs, that's why this was the reason why the Italian FFC decided to support this drug discovery project. Drug discovery project started in Genoa, where I worked, where we at IIT, we performed or carried on all the medicinal chemistry and in vitro DMPK activities, while still in Genoa at the Kids Institutet Gazzini Kids Hospital, they performed the in vitro screening of the compounds. So the story started with a phenotypic high-throughput screening, testing our internal small molecule library of around 11,000 compounds on 2 different cell lines. So this is a different paradigm for what I've seen so far. we tested compounds on 2 different cell lines, directly in cells. And we wanted, by doing so, to identify potentially novel compounds, novel correctors. So we carried on this isotope screening on these 2 cell lines, as I said, overexpressing Fabio and del CFTR. So what we did at that time, we identified few initial hits within 2 cell lines, and to keep a very long story short, through an iterative process of designing, synthesis, testing, and analyzing different drug likeness properties, we synthesized more than 400 compounds. We started here with this initial hit, and we identified a very potent efficacious compound, much more than the drug available at that time. So the compound is now named as 23765, from now on 765 for simplicity. As you can see here, this is the small molecule corrector, which shows a sub-nanomolar activity in rescuing the function of delta F CFTR in primary human bronchial epithelial cells, in primary cells, okay? Derived from patients with homozygous for this mutation. The compound is actually at the moment the preclinical stage and the preclinical stage development compound, in biotech, in an American biotech company. But nevertheless, what we have done in the last 3 years was to try to identify the target of this molecule, because as I mentioned, we tested our library and we identified this compound in cells. So we proved that the compound is indeed active in cells, but we didn't know for sure whether this compound was active directly on CFTR or eventually on other targets involved in the regulation of the trafficking and the rescuing of CFTR. So at that time, we started our activity with the aim in live cells, okay? So in an intact biological setting, to identify the target protein of 765 and possibly its mechanism side of action. One step back. We looked at by looking at the structure of the compound, we had to identify a chemical biology approach to somehow, link the compound to its biological, its more reasonable biological target. And by doing so, we identified this photo-affinity labeling technique as the proper one. So by these techniques, starting from the structure of the active compound, photo-affinity probes are designed and synthesized, featuring a photoreactive group and a reporter, chemical reporter functionality or purification tag. You will understand in a minute what I mean about that. So then we test the activity in biological, in the same functional cell where the active compound has been identified. And then we incubate our probes in cells, on cell lysates. So of course, these reasonably, the probes get in contact with the most proximal target, its biological target, and upon photo activation of this photoreactive group, a covalent bond is formed between the probe and the target, the biological target. Then put down, so identification of this protein probes adducts, and possibly identification of the target by different techniques. So starting from the structure of 765, we decided to use the diazirin. So this molecule here has a photoreactive group, which can, upon irradiation at 360 nanometers, form some very reactive chemical species, therefore forming a covalent bond with some biological targets. And this thiobiotin as a reporting target, something that we could use to fish out the adduct between the compound, the probes, and the target. So we proved the activity of our probes in the same fashion, let's say, where 765 was identified. And then we went to the lab and we tested the probes in intact cells, overexpressing both wild type and delta F CFTR. So we incubated them, we photoactivated, then we lysed the cells, and we using streptamidine beads, we pulled down our adducts, and we analyzed them using an anti-CFTR antibody. So what you can see here, these black spots, this one, represent indeed CFTR. So we could prove that our technique was helped to identify CFTR as the target, as a primary target of our class of compounds. And we also confirmed that by some competition studies where we tested here with the same study with an excess of the parent compound. So basically, you can understand that the excess of the compound can hinder the, let's say, same site as for the where the probe can interact with. So therefore, there's no evidence of the interaction. So there's no sign of this black spot. So having proven that, we also did that with other class of type 1 correctors. And as you can see here, there were all, in all cases, no sign of the CFTR. Therefore, we proved that they indeed all go to the same region within the CFTR. And likely, we also proved that type 2 and type 3 correctors, indeed, they showed some, the black spot corresponding to the interaction of our probe with the CFTR. They're proving that they go in at different sites. So this was already another hint that our compound might belong to the type 1 class of correctors. Then we went further. We knew now that CFTR is indeed our target. We wanted to understand exactly where this compound binds to. So we did we transfected X cells with CFTR fragments featuring either MSD1. MSD1 is a synonym of TMD1, as you have seen before. So transmembrane or transmembrane domain 1 and transmembrane domain 2. We transfected these cells, as I say, with these different domains, and we proved that indeed our compound enhanced the expression of MSD1 containing fragments, as does another type 1 of correctors. On the contrary, the compound does not have any effect in the expression of CFTR fragments on MSD2 counterparts. Furthermore, we also proved that in some cyclohexanoid chain studies where we stopped the synthesis of the protein for around 6 hours, we proved that the compound stabilized MSD1 containing CFTR containing fragments, as you can see here, and does not have any effect in MSD2 CFTR domain of CFTR. Then, so we have proven that CFTR is indeed the target. We more or less know that the compound goes at this side of within the CFTR, but now we wanted to get further into this research activity, and we performed some docking analysis based on some PDB structures reported in the literature, where which have been collected by some Curie studies on other type of correctors. We identify some key interactions of our compound within this pocket of CFTR, and we somehow prove that our compound goes indeed somehow in the same group where type 1 corrector 61 and 809 have been reported. We identify some key interactions, these salt bridges of the carboxylate group and these pi interactions of the aromatic parts of the corrector, and we did want to demonstrate whether these were indeed some key interaction within this putative binding site. So we performed some in silico mutagenesis scan, where we mutated some of these initial key interactions with some other amino acid residues. We ranked them and we selected some of them, and we brought them back to the wet lab where we performed some cycle-reactive mutagenesis studies. We transfected X cells with these double mutants, basically the F5 with the CFTR plus the other mutation that I mentioned. And we indeed proved that. So this is the single mutant, say, the activity of the corrector with a single mutant. These are the activities of the double mutants. You can see here that indeed these three key residues are very important and are within the putative binding site of our compound. So we also proved that the compound gets into CFTR and gets at that specific pocket. Sorry. So, lastly, we performed some molecular dynamic studies where we finally confirmed that 75 gets into this groove, this pocket where the other type 1 of correctors have been reported, and basically all the interactions, the key interactions have been identified through the docking studies have been also confirmed in these MD simulations. Specifically, these interaction between the tryptophan 361 with the aromatic portion of the benzodiazole of our corrector is the key interaction that brings our compound into this pocket. So I'm done. To conclude briefly, I hope I have given you an idea that by using this chemical biology approach and by following a different paradigm as compared to the one we have seen so far. So starting from an active compound, we went back to identify its target and its binding site. So we indeed proved that CFTR is the target of our corrector, and we proved also that it gets to this putative binding site. So last, a huge thank to the Fondazione per la Ricerca sulla Fibroagistica, which has supported not just the drug discovery project I mentioned earlier, but also all the activity I mentioned, and not to mention, of course, all the people that contributed to this work, primarily Francesco Sarcoliti for the chemistry part and Elisa Romeo for the biology. Then last, and all this, all of you for the kind attention. Thank you.
Thank you, Fabio. Very interesting talk. You have a question from Ijen?
Thank you very much for your intelligent presentation. Just a question in addition to what you said, which is very interesting on the stability and level of the protein synthesis. There is also some allostery modulator, if I understand, is a treat therapy, which is EVA factor. Is it, how does it fit with your data? Do you have any evidence for that? I'm sorry, it's not on your talk, but it's in addition to the treat therapy.
Yeah, I go back.
Actually,
you got a very good point.
In the market, if I understand right.
You got a very good point. Here we are. This one. Okay. So just to take a step back, this is a potentiator. Okay.
So it's an allosteric modulator.
Yes.
Yes.
So, and if I go back here, so this is the putative pocket where this potentiator has been crystallized in some cryo-EM studies. Okay, this is the potentiator. This is type 1 corrector. This BX8. No, this is 661 because, and this is the type 3 corrector. These three compounds all together they make the triple combination known as Trikanda. This is the medicament that patients are trying.
Are the correctors acting on the mature protein or on the
that's this is the very complex question.
biosynthesis?
Yes.
Of the protein in the.
There is a huge,
While it's clear that EVA factor is acting on the mature.
wonderful point. Yes. There's a huge debate. I've been to a international conference on CFTR modulators 2 months ago. There's still a huge debate whether the correctors, especially type 1 correctors, act co-translationally or post-translationally. There are publications saying one way, there are other publications say the opposite. The most reasonable explanation is that they act co-translation. So while the protein is produced, they interact with this nucleotide site and somehow they rescue the folding. Concerning the potentiator itself, they act at the end. So when the protein is already up to the plasma.
The compound is in which of the three?
Sorry,
Compound is in which of the three? In which one? Your compound?
type 1, here at this level.
You overlaid your compound with the other?
No, none. But we proved that it gets into this pocket, so reasonably it goes there.
Sonny?
I was very intrigued by your PAL combination with the biotin. I think it's quite neat. I just wanted to double check, how did you really get to decide where your exit vector, where you're going to place your exit vector, or you just synthesized multiple variants and you checked which one was still active?
Good point. Very good. I presented just, and I went quite fast. I presented just 2 probes, but as you can see here, the diazorin is placed into different portions of the molecule, and somehow also the biotin. So we indeed design and synthesize different probes, and here I just presented the ones that work. So this is as the drug discovery approach. It's a trial and error. So we design and test it. As long as the compound is active in the functional state, it's fine to be, taken over the next step on the photo-affinity labeling studies. So that's a good point.
Okay, so let's thank Fabio again.